My name is Robert Raffai. My affiliation is I work with the University of California, San Francisco, and the VA Medical Center in San Francisco.
My name is Bogdan Mateescu. I'm working in ETH Zurich in Switzerland.
Our research plan really builds on our accomplishments from the first phase of ERCC1, which was built around developing approaches to isolate carriers of extracellular RNA that comprise extracellular vesicles, plasma lipoprotein particles, as well as protein-associated RNA. And from these approaches that we've developed that oftentimes are considered now the gold standard methods, they require a lot of time. They're very user-dependent. The results can vary based on the individual that carries out the experiments. And oftentimes they require a lot of input material, a lot of biofluid. So the approaches during the last phase, the last few years of the ERCC1, actually Bogdan organized a supplement request building on ideas he had to try to streamline the methods for the isolation of extracellular vesicles and the other carriers. And he came up with a plan that we've integrated as a multi-PI project between himself, and our other investigators not present here, Dr. Kendall Jensen. And Bogdan, you may want to introduce the plan.
The plan for the next stage of the Extracellular RNA Consortium, the phase 2 of the program, is to answer different objectives. The first one is to reduce the sample volume that we can process to gain more sensitivity and more specificity in the xRNA-based assay for the clinical studies. So this is really the plan, and how we answer this is to develop new technologies that allow us to process very small samples. So as part of the proposal we have with Robert Kendall, and we implemented a new technological development, which is microfluidics, which allows to process and separate biofluids using different physical properties, but to process only very, very small volume, like let's say a drop of blood or a drop of another biofluid.
For me, it started through interactions with colleagues at UCSF. I was part of a multi-PI program project grant that developed at UCSF that was led by, at that time, Dr. Robert Blaylock, which sought to lay the foundations for the understanding of the biogenesis, the release, and the biodistribution of extracellular vesicles. And my participation in the group was that I had expertise already in the past in the isolation of lipid-like structures in the plasma and biofluids, so I brought an element of knowledge, and from that we developed methods. And I guess Bogdan approached it in a little bit different way.
Yeah, on my side, I am an RNA biologist primarily, so I'm getting interested into the process of regulation of RNA in cells. And I learned basically about extracellular RNA while I was actually investigating a process of cancerogenesis in France. And I was actually wondering how these processes were regulated, whether it was actually something active or like passive, how it could be useful for the patient in terms of clinical studies. And this is how we joined later the consortium by developing some very fundamental studies and starting from this to come back, and I'm very happy to come back to the clinics now, building on the fundamental knowledge that we developed over the last 5 years.
From my perspective, it'll be really tremendously useful to accomplish experiments that we will be proposing and we're funded to do, particularly the Department of VA, to investigate the role of extracellular vesicles, the role of extracellular RNA that distributes in biofluids of patients that are affected by severe forms of cardiovascular disease. So using this technology that we'll be developing as a group, it'll provide a level of understanding for prediction, for disease progression, disease treatment, and understanding the biology of how these RNAs participate in disease progression or resolution. Will, to me, that seems like it's going to make a big impact.
I think everyone has the same role, and we want to to make our discoveries useful for the patient and to offer the most robust approaches that people can use, which are not in the field can actually use in the lab and start using xRNA as a new tool to predict what it would be the response to a drug treatment, what will be the evolution of the disease in the next 3 years, 5 years. For me, xRNA will be one of the additional tools in the next years that clinicians can use to predict the evolution of the patients they are treating. So the idea is to provide a frame that we can improve the prediction medicine or personalized medicine approaches, and I think xRNA has a really good part of it because it's relatively easy, it's easy to implement, it's sensitive, and
can it be reproducible?
Yes.
Among centers.
Among centers, yes.